The electrochemistry of nerves. Electrochemistry is the relationship between chemical changes and electricity. Electrochemistry is most commonly applied to mechanics and metals, but it can also be applied to the mechanisms of the human body. One of the ways electrochemistry applies to the human body is in our nerves, and how they communicate with our brain and body. This video will examine the process of how nerves convey impulses, a process that involves electrochemistry, and the journey of the discovery of the mechanisms of nerves. The journey of the discovery of the mechanism that allows electricity to influence nerves and muscles began in 1730, when Stephen Gray gave a boy a charge of static electricity. Over the next 50 years, it was observed that contact with an electrically charged rod was able to cause the muscles of a dead animal to contract. Luigi Galvani was a major contributor to the discovery of how electrochemistry applies to nerves. In the late 18th century, Galvani began to experiment with electricity and muscles, and by 1791 he had discovered that by attaching a metal arc to a frog-legged muscle, he could make the leg kick without adding electricity. In the late 1840s, Émile de Bois-Reymond used sensitive new equipment to show that electrical currents flowed through muscles and nerves, and changes in the intensity or direction of the current caused the muscles to contract. Later that decade, doctors began experimenting on live patients and were able to make individual muscle fibers contract, resulting in more detailed maps of the muscular system than was previously available. In 1902, Julius Bernstein proposed the membrane theory, the first believable physicochemical theory of bioelectrical occurrences. The theory assumed that nerves had selective permeability and were chains of concentration circuits. The theory gave evidence to assume that the electrical potential of the lesioned muscle is caused by the electrolytes, in particular by inorganic salts such as dipotassium, hydrogen, phosphorus, and tetraoxide, already contained in the undamaged muscle fiber, Bernstein stated in 1902. In 1938, David McManson demonstrated that polarization travels along the nerve to the end, where it triggers the release of the chemical acetylcholine. Adjacent neurons become polarized by the chemical, and the impulse travels towards its destination, whether that is a muscle, another nerve, or an organ. The chain of chemical reactions breaks down the molecule adenosine triphosphate, also known as ATP, releasing electrochemical energy. At one end of a nerve cell, the nucleus is surrounded by the soma, with dendrites branching out around it. A chain-like structure called the axon branches off the soma. The axon is broken up into Schwann cells, covered by myelin sheaths, and separated by nodes of Ranvier. At the end of the axon, the cell branches out into axon terminals. Neurons are surrounded by a plasma membrane that separates the intracellular and extracellular fluids. The extracellular fluid is positively charged with respect to the intracellular fluid, and is measured to be about -50 to -100 millivolts different in animal cells. This difference is known as the membrane potential. A neuron at resting state, when it is not transmitting electrical signals, has a membrane potential of around -70 millivolts. The intracellular fluid has a high concentration of positively charged potassium cations and negatively charged anions, such as proteins, amino acids, sulfate, phosphate, and other anions. It also has a low concentration of positively charged sodium cations and negatively charged chlorine. In the extracellular fluid, the highly concentrated cations and anions in the intracellular fluid exist in a low concentration, and the lowly concentrated ions inside the neuron are highly concentrated. Electrochemistry in nerves is evident in the way ions move between the intracellular and extracellular fluids and through the plasma membrane. The plasma membrane has selective permeability that allows dissolved ions in the fluids to pass in and out of the neuron. Ion channels in neurons are aqueous pores made of specific transmembrane protein molecules that allow selective ions, such as sodium, potassium, and chlorine ions, to cross the plasma membrane. Neurons are mostly permeable to potassium because potassium-permeable ion channels are more abundant. Because of the membrane potential, the high permeability of the plasma membrane to potassium ions concentrated inside the neuron flow out of the neuron in an efflux. The positive charge of the potassium increases the negativity of the intracellular fluid, increasing the difference between the charge of the inside and outside of the neuron. The increased negativity attracts an influx of charged potassium back into the neuron. The plasma membrane is also permeable to sodium. The negativity of the intracellular fluid attracts an influx of sodium ions from its high concentration outside the neuron. Without sodium, the membrane potential would be negative 85 millivolts, but with sodium, the membrane potential becomes negative 70 millivolts, as some of the positive charge leaving the inside of the neuron with potassium is replaced with the positivity of sodium entering the neuron. The sodium-potassium pump is a plasma membrane protein abundant in neurons that uses ATP to transport sodium out of the neuron and potassium into the neuron to restore the concentration gradients of the ions in the intracellular and extracellular fluids. Neurons and muscle cells are excitable cells and are the only cells that can generate changes in their membrane potentials. Neurons generate changes through gated ion channels. These channels respond to stimuli, triggering hyperpolarization and depolarization. Hyperpolarization is an increase in the electrical gradient across the membrane that occurs when potassium channels open, creating an outflow of potassium. This makes the relatively more negative intracellular fluid more negative. Depolarization is a reduction in the electrical gradient across the membrane that occurs when sodium channels are opened, creating an inflow of sodium. This makes the neuron less negative. The voltage changes that occur during hyperpolarization and depolarization are called graded potentials. The stronger the stimulus, the more channels will be opened and the more impulses will occur in a shorter amount of time. During depolarization, the voltage of the membrane potential can increase by about 15 to 20 millivolts, from the resting potential of negative 70 millivolts to about negative 50 to negative 55 millivolts. This is the threshold potential, and it triggers an action potential. An impulse is created by an action potential. Sodium channels have two voltage-sensitive gates, an activation and inactivation gate. The inactivation gate is open at resting state while the activation gate is closed. Depolarization stimuli influences the activation gate to open quickly, allowing a large influx of sodium to enter the neuron. The inactivation gate closes slowly, cutting off the flow of sodium. The repolarization phase involves the potassium-gated ion channels opening their only gate and allowing potassium to exit the neuron. This gate is slow, so when it closes in the undershoot stage, the neuron is more negative than it is at resting state. Action potentials occur at a specific point in the neuron and trigger the neighboring region to become depolarized, reach the threshold potential, and for another action potential to occur. A chain of action potentials occur and travel along the axon to the axon terminals. Parts of the nerve that have already reached an action potential cannot be stimulated again because the inactivation gate of the sodium ion channel is closed after repolarization, in a period of time known as the refractory period. The neuron can only be depolarized when the inactivation gate opens again. The neurons of vertebrates have developed saltatory conduction, the mechanism of myelinated axons that concentrate ion channels at nodes of Ranvier, so that the action potentials occur there and trigger action potentials at the next node of Ranvier. This makes impulse transmission faster. Thicker axons also increase the nerve's ability to quickly convey an impulse. Impulses are able to be passed from one neuron to another across electrical and chemical synapses. At electrical synapses, a gap junction allows an impulse to flow directly from the presynaptic cell to the postsynaptic cell. At chemical synapses, the impulse is converted into a chemical signal that travels across the junction, and when reaching the postsynaptic cell is converted back into an electrical signal. It is evident that the processes of nerves vital to the functioning of living beings involve electrochemistry, and the journey in the discovery of the relationship between nerves and electrochemistry is a contemporary discovery.